everybody, I'm Tall.
And I'm Small.
And today we're doing Khan Academy's MCAT Prep Cells Section Cellular Division questions. First question. Oogenesis, the process that creates female gametes, is halted at prophase 1 until puberty. Which of the following describes the DNA content of a female's gametes during her childhood? So how do you describe the DNA content during prophase 1? 23 chromosomes and 23 chromatids, 23 chromosomes and 46 chromatids, 46 chromosomes and 46 chromatids, or 46 chromosomes and 92 chromatids. So if you're at prophase 1, then you have duplicated everything. You haven't undergone meiosis 1.
Yeah, there's been no splitting of anything. You're just now forming your spindle and starting the prep for everything.
So if that's the case, then wouldn't that mean... I'm notoriously bad with this, but if you're at prophase 1, I feel like you'd be at 46 chromosomes.
Yeah, so we start with a full count of everything. Mm-hmm. And then especially if you doubled everything, 46 would make more sense than 23. But we know that meiosis, that whole process, is a reduction division. So I was expecting them to say something like, like at some point later on what would happen. And I think they're waiting for us to kind of assume that. But it's good that we recognize, prophase 1, nothing's happened yet. We've just duplicated and that's it.
Okay. So chromosomes, I understand 46. I don't think I know what a chromatid is. Is that half a chromosome?
Yeah. They're just the different arms.
Okay, so it should be 46 chromosomes and 92 chromatids.
Yeah.
And we got it right.
Nice. Let's read the hints, though.
Okay.
Prophase 1 is the first step of the division of meiosis, which we knew. Mm-hmm. A chromatid is a single copy of genetic information, while chromosomes are counted by the number of centromeres. Okay. In prophase 1, the cell has undergone DNA replication, but has yet to divide, leaving it with 46 chromosomes and 92 chromatids.
So a chromatid, it's like when you see the typical X-looking chromosome, one half of that X is the chromatid, which carries one allele, but you have two alleles, two chromatids for every chromosome.
Yes.
Okay.
Exactly.
All right. Bio 1 stuff, but I'm relearning.
So that 23andMe thing, it gives you all, what does it give you? But it's for all of the 23 chromosomes?
Chromosomes.
Right.
So technically they're giving you, they're analyzing 46 chromatids.
Right.
Because each chromosome is going to have two alleles, which will, matter for your phenotypic expression of your genome.
Nice.
Very cool. Next question. Oogenesis and spermatogenesis describe the process of meiosis in females and males respectively. What statement about the two processes is true? Oogenesis forms two polar bodies, while spermatogenesis only forms one. Both are halted at an intermediate step until puberty. Oogenesis produces a larger gamete than spermatogenesis, or both produce the same number of viable gametes. Okay, so let's process of elimination this, because I think I already know what the right answer is.
Right.
So both produce the same number of gametes.
Yep.
What's up with that one? I know I'm making lots of gametes all the time.
Yeah. You are making, what is it, like 60,000 a day or something crazy like that?
That's like normal people. I'm more like 100,000.
Mm-hmm. Okay. Then that would be all the testosterone, ladies and gentlemen. Okay. So I'm going to pick a different one that I want to talk about.
Okay.
Forming two polar bodies in oogenesis versus only forming one in spermatogenesis. I don't think you guys form any polar bodies.
We don't. We utilize all of our products of meiosis too.
Okay, but I'm just going to go ahead and say that your products of meiosis too are a little bit inferior, as they basically are only delivery vessels for a Y chromosome.
And we're back on this again.
Okay.
But so yeah, two polar bodies, oogenesis. It undergoes meiosis too, but one of those things becomes just a kind of like dead polar body.
Yeah, so it happens in meiosis 1 as well, right? So we have our oocyte.
Something like that.
The naming of it changes as you go down the differentiation. Mm-hmm. But as you go down, you make two polar bodies, I think, and then one viable egg. And it kind of makes sense, right? So sperm can all be equally as small or large or whatever they need to be to carry the basic necessities of what they give to the gamete. To the zygote. But the egg needs to be really big. It has all of the nutrients. It has a bunch of the organelles.
The mitochondria.
Yes, you get your mitochondria from your mom too. So the egg needs to be bigger. So it kind of makes sense that we shirk on the other two we make that are just useless and get degraded. And put all of our eggs in one basket, so to speak.
Put our mitochondria in one egg. So, yeah, so that would lead us to oogenesis produces a larger gamete than spermatogenesis.
Yes.
Spermatogenesis, all we need is, like, basically a rocket propulsion and the DNA.
But they're bad at it. They don't ask for directions.
They don't. Okay, so next question. Question number 3. Which of the following is not a reason cancer is hard to treat? Increased mutation rate, increased growth rate, increased metastasis, increased contact inhibition. Which is not a reason cancer is hard to treat. So let's find all the reasons why cancer is hard to treat. Increased mutation rate. Yeah.
Well, I don't know, right? Isn't it just the first one and that's what messes it up?
I don't think so.
Because it just needs something to mess up surpassing a checkpoint. And then it's gone.
So like theoretically then, there are multiple things that could cause a cell to become cancerous, like multiple genes.
Yeah.
So if you have one that mutates, wouldn't 2 that mutate make it more difficult to treat?
I'm sure. I'm not quite sure what contact inhibition is.
I think that's when like if you have 2 growing things that butt up against each other, they know to stop.
Oh, okay.
So I just said growing, so that makes me think that increased contact inhibition. So increased inhibition, that doesn't sound like cancer.
Yeah.
Metastasis, definitely.
Yep, increased growth rate, definitely.
Growth rate, definitely. That's how it like makes the problem. So yeah, I think even just looking at the words increased inhibition, that doesn't sound like cancer. And we were right.
Yeah.
Okie dokie. Question number 4. During which phase does the nuclear envelope begin to disappear? Anaphase, S-phase, cytokinesis, prometaphase. I think it's before you even get into mitosis.
Oh, really?
Yeah, so I don't think it should be anaphase, cytokinesis, or prometaphase.
Anaphase seems a little late.
It does seem a little bit late.
We're already lined up at the plate in the middle of the cell. Cytokinesis, definitely not.
Right. Prometaphase, I don't think so. I mean, that's just like, that's between
Prophase and metaphase. I would almost go with prometaphase.
Really?
Yeah, because I just remember hearing, G1, S, G2, G1 and G2 were like growth and doubling of organelles and prep for everything. And S was just doubling of the genetic material.
Right.
So I never remember hearing something about nuclear. I just don't remember.
Yeah. If they had said G2, I totally would have picked that. But I think I gotta go with it happens before mitosis, so I gotta say S phase. All right.
Oops.
Oh, wait, I was wrong too. Hold on.
Oh, yeah. It is prometaphase.
Oh.
Yeah. The nuclear envelope is responsible for protecting the genetic material from the rest of the cellular contents. That's pretty important. In anaphase, chromosomes move to the opposite sides of the cell, like we said. In order to allow for proper metaphase alignment and chromosomal division, the nuclear envelope dissolves in prometaphase. So it didn't really explain it. But.
Okay, so they're basically just saying that in prometaphase, that's the point when we need the chromosomes all to leave the nuclear envelope?
Yeah, we're about to start shipping them to the middle of the cell, so that makes sense. They don't really have to pass that gate until we're done with prophase.
Okay, that makes sense then. All right, question number 5. Centrioles can be found in both eukaryotic and prokaryotic cells, only prokaryotic cells, only eukaryotic cells, or in eukaryotic cells and in prokaryotic cells undergoing genetic recombination. Centrioles. Centrioles are the dudes throwing out those microtubules to then pull apart the chromosomes.
Yeah.
So, I think they're found... Ooh, wait a minute. I almost fell into a trap.
Did you? What was the trap?
Prokaryotic cells don't have multiple chromosomes. I don't think they have anything. They don't do recombination.
I don't think they do, mitosis like that. I think but their version binary fission, right?
Yeah.
Binary fission is a different process.
Okay.
Like rolling strand replication and all that happens differently.
Yeah.
And now, to come to think of it, I don't know that I could really explain in detail what happens.
In my head, I've always just thought they synthesize the new strand. They have 2 strands, and then there's got to be something that moves them apart. But there's definitely no recombination phase. They don't line up on a plate.
No nuclear envelope reforms since there's no nucleus to begin with. Okay.
Yeah, that would make sense. So then we would say centrioles can be found in only eukaryotic cells?
Wow, I would have probably made that mistake unless we had talked about,
Yeah.
Why isn't the MCAT a group test?
Yeah, I know. Okay, next question. Which statement best describes chromosomal movement during mitosis? Chromosomal movement during mitosis. Microtubules bind tubulin polymers on centromeres to initiate homologous chromosome separation. Centrioles on chromatids bind kinetochores to raise sent by the asters. Oh, that's a word I haven't heard in a long time. Mm-hmm. Centrioles send out asters which bind to kinetochores to pull chromatids apart. Each centrosome binds an individual chromatid to ensure equal division. Okay, so centrioles send out asters which bind to kinetochore. So whatever aster is, I'm guessing that's what it means by like the microtubule formation.
Yeah.
I guess. They do bind to kinetochores.
Yeah. So that D answer, centrioles send out asters which bind to kinetochores to pull chromatids apart. That seems right to me.
Okay. I like that one. Let's look at the others.
Because at first I liked the initiate homologous chromosome separation. I was like, not meiosis.
Mitosis?
Yeah.
Ooh, homologous chromosome separation. So that never happened.
Yep.
So each chromosome binds an individual. I'm liking our centrioles send out asters which bind to kinetochores and pull chromatids apart. Okay, we got it right. Let's look at the hints real quick because I'm not totally solid on this. The kinetochore is a protein structure centered around a chromosome centromere. Centromeres are found on chromosomes. Centrosomes are found toward the poles of cells during cell division. Centrosomes are over by the centrioles then?
That's what I think.
Okay. Asters are microtubules sent out by centrioles to bind to kinetochores and pull microtubule. Okay. All right. So it was exactly what we thought. Okay, next question. Retinoblastoma protein, RB, is an important cell cycle checkpoint protein that prevents the transition from G1 to S. The presence of the cyclin CDK complex releases it from the genes that it binds. Which of the following mutation scenarios is least likely to lead to the formation of a neoplasm? A neoplasm. I think that's a cancerous thing, right?
It sounds like if I'm surpassing some sort of checkpoint, then yes.
Okay. Okay, so possible answers. Oh, so which of the following mutation scenarios is least likely to lead to the formation of a neoplasm? Loss of functional RB and no CDK mutation. Loss of functional RB and CDK that does not respond to negative feedback. No RB mutation and CDK that cannot bind to cyclin. No RB mutation and CDK that does not respond to negative feedback. So CDK, if you have CDK and it's active, it releases the RB complex and allows the transition from G1 to S.
Yeah.
So more CDK stuff, more cancerous.
Yep.
So no CDK mutation, that doesn't sound right. CDK that does not respond to negative feedback, that sounds pretty good. CDK that cannot bind to cyclin, that doesn't sound right. So it's got to be...
But the cyclin CDK complex releases RB from preventing, G1 to S.
Yes.
So if CDK can't bind to cyclin, then it won't release the thing that's checking the growth.
Right, yeah. So
least
that
likely to lead to the formation of neoplasm.
Well, so I'm kind of guessing here that neoplasm means cancer.
Okay.
So if you can't release RB, then you can't move from G1 to S. Therefore, you don't grow.
Right.
So it wouldn't lead to cancer.
Right.
Okay, yeah. So it's got to... I think that it's got to be CDK that does not respond to negative feedback. Therefore, it's just going to keep going.
That is... Well, Which of the following mutation scenarios is least likely to lead to the formation of neoplasm? Keep growing is creating the neoplasm.
Oh my god, I was reading this very wrong.
Yeah.
Okay. CDK that cannot bind to cyclin. So, So, loss of functional RB and no CDK mutation. So, CDK mutation would possibly be good, but if you don't have functional RB, then that's going to bypass that whole thing.
Mm-hmm.
And your answer, which was no RB mutation and CDK that cannot bind to cyclin, means that you are probably not going to form cancer, as I was just saying, but totally not. Like my landing zone was not cleared for
landing
for that answer to come and land on me. And okay, we were right. You were right. All right, next question. A karyotype can be used to check for certain chromosomal mutations. In what cell cycle phase can cells be karyotyped? So it's used to chromosomal mutations.
Mm-hmm.
S, G0, metaphase, prophase. In what cell cycle can cells be karyotyped? Chromosomal mutations. So which one of these things is not like the other? If we're in metaphase, they're out of the nuclear envelope.
Yeah.
Every other one, they're in the nuclear envelope. So that would be my thought process and why I would answer that.
Okay.
What are you thinking?
I wasn't really sure. I have a hard time believing that it's any of the G's and the S. I think they might not have condensed into chromosomes yet.
Mm-hmm.
So I was thinking either prophase or metaphase, but I really don't know where to go from there.
Okay. So prophase, we learned just earlier that prophase means it's literally just the nuclear envelope thing, I guess, that I'm thinking.
Okay, I like your reasoning. We'll go with metaphase.
Hey, okay, we got
Nice.
it right. What do those hints say? Karyotype is used to visualize separate chromosomes. Chromosomes do not begin to condense until prophase. Karyotype shows DNA as pairs of X's. Sister chromatids split in anaphase, meaning at a karyotype. So you'd have to be arrested in metaphase, or else you wouldn't be able to do a karyotype. Okay. All right. Next question. Question number 9. Colchicine, I think that says colchicine, is a drug used to treat gout. It also happens to arrest cells in anaphase. Which cytoskeletal protein does colchicine affect? So it arrests them in anaphase. Is it myosin, actin, keratin, or tubulin? If it's an anaphase, then we've pulled apart already, right? Or
we're in the process of pulling apart.
So they're maybe saying that you can't pull apart? Well, we know that tubulin...
Well, it says they're already in anaphase.
Mm-hmm. Okay. So we know that tubulin and centrioles, they're used in metaphase to line them up.
Yep.
So that has to be working.
Mm-hmm.
Keratin, I don't think that has anything to do with cytokinesis. Actin and myosin, they do... one of them's got to be involved, I think.
So my thought process was the microfilaments, film actors, myosin, all those.
Right.
My synopsis on those from the other ones that we looked at were, endocytosis, exocytosis, and movement, like amoeboid movement.
Mm-hmm.
And I'm not talking about that in here. So I didn't really know what keratin was, but tubulin, a microtubule.
Yeah.
I know that microtubules are what helps us elongate in mitosis, so I went with that.
Okay, and that was right. Let's see. Anaphase is the stage where sister chromatids are pulled apart. Okay, so, microtubules from centrosomes bind chromosomal kinetochores to separate the genetic material, and microtubules are made from tubulin, a protein that can be bound to... Okay, so it inhibits tubulin polymerization, prevents microtubules from being formed. I don't know if I totally love that, but I guess it makes sense because the other ones aren't involved in that process. Okay, last question. Which of the following would you expect to see in a cancerous mass? Decreased ATP use, increased time in G0, gain of function mutations in checkpoint proteins, loss of function mutations in apoptosis controlling proteins. So I would assume if you're growing a lot, you're using a lot of ATP. G0 is the phase where you're not doing any kind of cellular division.
I think so, yeah, that dormant phase.
Gain of function mutations in checkpoint proteins, I think it's always loss of function.
Yeah, it sounds like the opposite of that.
Right. And then we have loss of function in apoptosis controlling proteins. So you don't do cell-induced death, and it's allowed to just keep growing even though it should be doing apoptosis.
Sounds good to me.
And we got it right. All right, very cool. I got that one really dumb one wrong, but strong work either way.